Welcome to this presentation on artificial intelligence and the design of experiments in the development of pharmaceutical formulations, a taxonomy of approaches. This presentation is based on research conducted by Michal, HOFFMAN and Marcin MRUGALSKI from the University of Zielona Góra in Poland. Our main goal today is to provide a clear and detailed look at artificial intelligence methods and explore how they can be used to develop new pharmaceutical formulations. Throughout this presentation, we will talk about the strict limits of traditional methods, known as the design of experiments, which are normally used when making pharmaceutical formulations. We will also point out the potential ways to get past these old limits by using artificial intelligence. Finally, we will show specific artificial intelligence methods that are used to design systems that safely deliver these medicines into the human body. Let us take a brief look at the agenda for this presentation. The information is organized around 5 main topics. First, we will compare traditional design of experiments with artificial intelligence-based approach. Next, we will explore the different ways how artificial intelligence is applied in the actual design of pharmaceutical formulations. Next, we will discuss smart drug delivery systems that are created to safely pass through natural barriers inside the human body. In the subsequent part of presentation, we will present a taxonomy of the artificial intelligence methods used in the design of experiments. In particular, the area of application of the discussed method will be presented. Finally, we will finish the presentation with a summary of the most important findings and look at what the future research directions. Artificial intelligence is relatively often used in planning traditional processes or products, but sporadically in pharmaceutical production. The limitations on the use of artificial intelligence methods stem mainly from the pharmaceutical industry's considerable conservatism regarding the generation of pharmaceutical formulations using techniques that lack determinism, predictability and transparency. Now, classical design of experiments methods based on linear regression are frequently used to the development of pharmaceutical formulations, but they have major limits. They can generate biased results when certain assumptions of the linear model are not met: linearity of the relationship between variables, randomness of the sample, independence and lack of collinearity of variables, as well as the assumption of constant variance of errors or noises and normality of the distribution of errors and noises. To solve such problems, it is possible to use methods belonging to artificial intelligence, among which we can mention artificial neural networks, neurofuzzy networks and Takagi-Sugino approach. These methods are gaining popularity since their use is possible in the case of modeling nonlinear dependencies even in the absence of knowledge of the physical laws describing the functioning of the modeled system. The application of AI methods in pharmacy allows us to move away from traditional analytical chemistry methodologies which rely on a laborious, empirical search for molecules in favor of automated, mathematical, multi-criteria experiments aimed at identifying new pharmaceutical formulations. Among the key areas in AI methods enabling pharmaceutical formulations design, the approaches described below can be highlighted. Virtual screening and molecular docking performed using global optimization. These algorithms efficiently search through energy states to avoid getting stuck in local minima during molecular docking, which prevents false results when testing how a drug connects to a receptor. Generative pharmaceutical formulations designed based on the deep learning. These models learn the rules of chemistry from huge datasets and use them to build entirely new, ready-to-make chemical structures from scratch. Multidimensional modeling of toxicology and ADME profiles using the perturbation theory machine learning. This method acts as an early safety filter before testing on living subjects. It actively decodes hidden toxic properties based on simple parameters so scientists can remove harmful parts early on. Lead optimization uses Bayesian optimization. This method gently improves promising compounds step by step to minimize design time and save valuable clinical trial resources. In the case of smart drug delivery systems, medicine must safely overcome natural physiological barriers to reach the 6 Ls. One major step forward is designing smart nanocarriers using deep learning. Older carriers often release their medicine all at once in a burst, which is very toxic to healthy organs. Smart nanocarriers undergo degradation triggered only by specific signals, like the acidic environment found around a tumor. This prevents the dangerous burst release and completely avoids hurting healthy organs. Another important tool is parameter prediction using predictive machine learning. These algorithms accurately forecast the perfect size of a liposome and its safety profile before it is even built. Finally, the scale-up process uses the adaptive design of experiment method. This helps minimize biological waste in highly complex treatments, like CAR T-cell engineering, by finding the absolute best combinations of variables for growing the cells. In this slide presents the taxonomy of artificial intelligence methods and the design of experiments for the development of pharmaceutical formulations. As can be seen, such approaches can be divided into 5 main groups such as predictive machine learning, deep learning, adaptive design of experiments, global optimization, and generative and support AI. Over the next 5 slides, we will break down exactly which algorithms belong to which group and explain their specific jobs in the development of pharmaceutical formulations. Predictive machine learning comprises a set of artificial intelligence techniques that predict unknown future events based on the analysis of historical data. It is based on the creation of approximation models, the purpose of which is to replace computationally expensive and time-consuming simulations with a simplified mathematical approximation. These approaches can be divided into 4 main groups such as artificial neural networks, support vector machines, random forest, and XGBoost. Artificial neural networks act as advanced models that map out the complex, nonlinear relationships between what goes into a process and the final quality of the product. Support vector machines are used for the classification of formulation stability. They help determine the exact boundaries between stable and unstable chemical systems. Random forest algorithm is used for variable significance analysis. This simply means it determines which factors have the biggest impact on how well the whole process performs. Finally, XGBoost serves as another substitute model to map out complex experimental spaces. It helps narrow the focus of the study to only the most important variables, saving a lot of time. Deep learning models learn from training data, automatically discovering relationships without the need to manually define features. These approaches can be divided into 4 following groups: multilayer perceptron, graph neural networks, recurrent neural networks, and transformers. Multilayer perceptron is used to model the link between many different process variables and the critical quality features of the final product. Graph neural networks allow for a direct representation of molecular structures as visual graphs. In these graphs, the atoms act as connecting points, and the chemical bonds act as the lines between them. Recurrent neural nets are effective at modeling data representing time series and dynamic systems. This is heavily used in studies to see if a drug stays stable over a long period. Finally, transformers, like the one called MolBERT, act as smart screening filters. They identify the most promising molecules long before the physical laboratory work even begins. Adaptive design of experiments involves sequentially selecting the most informative subsequent experimental points based on information from previous experiments, thereby maximizing the knowledge gained for the model. These approaches can be divided into 4 following groups: Bayesian optimization, Gaussian processes, Thompson sampling, and methods such as BALD, badge or corset. Bayesian optimization provides smart planning for experimental sequences to minimize the number of lab tests needed. Gaussian processes are used to estimate expected values and figure out how uncertain a prediction might be. Thompson sampling is a method used for picking the next best formulation variants. It does this by taking samples from the results the model has already produced. Finally, strategies like BALD, badge, and corset represent specific ways to select only the most informative and helpful data points for the experiment. The global optimization methods include heuristic and meta-heuristic algorithms, which are used to search for the global optimum in complex, multimodal solution spaces. They can avoid local optima and have the advantage of being able to search for large decision spaces. These methods allow us to both optimize the design of experiments and find optimal parameter values for the systems. This class of methods includes a large group of stochastic algorithms or those inspired by biological phenomena. For example, they include the following methods: adaptive random search, ant colony optimization, and simulated annealing. Adaptive random search method can be used in random search for drug parameters to maximize a predefined objective function. Ant colony optimization solves problems in chemical synthesis routes by copying the natural behavior of an ant colony searching for the best path. Simulated annealing method focuses entirely on searching for the global optimal result within complex, multidimensional models. Generative and support artificial intelligence methods enable models to be fine-tuned and allow machines to operate in a more intelligent and human-understandable manner. Generative methods make it possible to create entirely new products that are not present in the training database. They also allow for the creation of synthetic data, which enables models to be fine-tuned when there is a shortage of real-world data. These approaches can be divided into four following groups: generative adversarial nets, variational autoencoders, reinforcement learning, and fuzzy logic. Generative adversarial nets are responsible for creating completely new, synthetic chemical structures and images that are then used to teach other artificial intelligence models. Variational autoencoders focus on mapping molecular space onto vectors. This allows the system to generate molecules that have very specific properties that the scientists want. Reinforcement learning supports the generation of entirely new structures and optimizes complex, multi-step technological processes. Finally, fuzzy logic is a helpful method for turning human words and concepts into exact mathematical values using membership functions. To conclude the presentation, it should be noted that the combination of artificial intelligence methods and design of experiments in pharmaceutical formulation design appears attractive because of the ability to perform simultaneous multi-criteria optimization during their design. Goal-oriented optimization artificial intelligence-based tools can quickly balance a molecule's physicochemical parameters alongside its toxicological profile and synthesizability. Moreover, application of artificial intelligence and design of experiment in pharmaceutical formulation design allows for prediction of hidden nonlinear interactions. Classical statistical systems fail when confronted with the multidimensionality of the immune system's biological networks. Neural networks can predict with a high degree of probability that a given protein-based pharmaceutical formulation will be rejected even before it is translated into the genetic code of the protein being tested. Thus, the application of artificial intelligence and design of experiments in pharmaceutical formulations design represents an interesting direction for future research. Thank you very much for your attention.